good morning, good afternoon, or good evening, depending on where you are joining us in the world. My name is Brian Anthony of Mechanical Engineering. I'll be your host today, if you will. But wanted to say just thank you for joining our very first MechE Alliance Seminar Series. In this unusual environment in which we're all working, we find it an opportunity, but also important to make sure that we're engaging our community at MIT and the department and worldwide. So we'll have a series of talks from MIT mechanical engineering faculty, from students, and from partners. And it's a great pleasure to have our first speaker with us today, Professor Mathias Kolle, Professor of Mechanical Engineering, has interest in tunable optical materials, manufacturing of optical materials. He's been faculty at MIT since 2013, did his PhD at the University of Cambridge, and has some exciting work to share with you on how to manipulate light and color with both soft and structured matter. So let me just stop my sharing and I'll turn it over to him. But also as we go, keep in mind that we will have a Q&A at the end. There's a Q&A function, so if you can please type your questions in there or raise your hand with the Zoom raising hand function, and we'll at the end sequence questions in the order that they came. And with that, I'll stop sharing and turn it over to Professor Kolle.
Brian, thank you. And Teresa and Brian, thank you for having me as a speaker in the MechE Alliance. This is quite an event, I think. There's 41 participants, or 45 now. And unfortunately, I cannot see you all. So I'm going to speak into my screen. And that means that at some point, I will do a sort of check as to whether I'm too fast or too slow. And if you can give me some feedback, that would be genius. Now, I think I figured out how to share screen, and you should see the first slide right now. The topic is manipulating light and color with soft and structured matter. And you can see that I want to cast this a little bit in a sort of lunch seminar type style. And so I cooked up this big cauldron of stuff that is cooking there and simmering, showing different colors. And without further ado on that, I want to present the menu for this lunch seminar. So I'll start off with a couple of hors d'oeuvres if you want, some intellectual palate teasers. Unfortunately, I cannot give you the real food. Also, I also think that what I cook up, you probably won't like to eat at this point. And then I want to start with starters, some research overview. I want to present my team and do a short flyby with butterflies. And in the main course, as said in the abstract, I will talk about colorful materials, fibers and droplets, if I have time. And for dessert, we have questions, and I hope you have some. All right. Now, for the hors d'oeuvre, there are three different things that I can try to speak about today. One is butterflies briefly and how to see biology make materials. The second one I want to point out is seeing how knots and tangles in color show their mechanics. And the third one, if I get there, is looking at the colors and the rainbows in micro droplets. This is sort of the palette teaser. I have another palette teaser. It's a bit of a playing around thing of mine. I usually think you should always over-explain a slide. And today I want to try to see if I can under-explain a slide. And I just want to show you a couple of really cool pictures from a piece of work that Cecile, who is in the group of John Hart now, but was a visiting student with me a while ago, has just recently published in Nature Photonics. So here are these wonderful sets of data. We can see biology with this sort of technique that Cecile has developed, which we call SLAT imaging, very nicely. And I'll leave it exactly there. So you'll have a lot of questions, I hope, at the end, if you want to know more about this technique. So that's the second palette teaser. And with that, I'm going to go into starters. And I have actually not started my stopwatch, so I don't know what time I have. Let me do this right now, so I'm not running over too much. What I've cooked up for you today is sort of a mix of things from not only my group's cookbook, but we also take a lot out of the cookbook from nature. There's really interesting ways of manipulate light and play with colors and play with other effects. And the ingredients that we like to use in my group for making these sort of interesting light manipulation concepts real are soft and fluid matter, because you can change them, you can tweak them, you can push them and squeeze them. And if you structure them correctly, that will change the optics. And if we do this right, then hopefully we cook a couple of interesting things that are actually useful, not necessarily for eating yet, but for other sort of applications like sensors and beautiful color displays. Now at this point, I'll pause the sharing for a second. And I'm going into slightly different presentation mode, which I haven't really done before. So that will take a moment. because I want to point out the cooks that actually have put this menu together for you. Can you see my background now with all these amazing people? Teresa, I need a pointer from you.
We see it behind you in the small window, but your slide is still showing on the main
Slide is
screen.
still showing. Okay, good. Good.
There we go. Now I see it.
Perfect. Thank you. So these are really the chefs behind everything that I show you today. So I'm squeezing out of the picture for a second. You can see a couple of different names here. The first names, and I show you the last names as we go along the slides. They're all really good in different parts of the work that we do. And I'd like to say something that is a bit of a complaint to some extent about the professorial position. Often it's this guy in front of the screen, in front of the audience, that sort of gets into the bullfrog seat and then sort of makes a big position and a big statement. But it's really a lot of people behind that cook everything that this guy speaks about and sort of play the music for me in the bullfrog boasting seat. So I can only do that bullfrog boasting because of all my students and the stuff that they're really doing very nicely. And so my plea in that context is that, or my credo is that if I ever become too bullfroggy, I should remember that I have a whole concert behind me that actually makes the music for me to boast about. And the work that I'm going to point out today in particular is the work from Anthony. And Anthony is there, and Sarah, and also from Joseph, who is here. This is really hard. All right, I'm going to change back into my share screen mode. And you should see my slides now. You have the same one. And I forgot to point out Ben, because he has also done some really nice thing that I show a slide about. All right, we start with Anthony's work. Anthony has figured out how to keep the whole life cycle of butterflies going in my lab. And with this whole life cycle of butterflies, you can see that starts from the egg and from the caterpillar, but then goes into the chrysalis, where the whole butterfly goes through the stage of metamorphosis where it develops the final imago, or the adult stage that you see on the top right. This is a great model system in our point of view to study biomechanical processes and to understand how structure forms. We're not the only ones. There's people across the world, in Europe and also in Asia and Australia, that have interest in this. But what Anthony is doing is he does a very cool thing. He takes the butterfly and he takes it in the chrysalis that you can see in this picture here, and makes a small window in that chrysalis. And that window is not detrimental enough for the butterfly to not make it. It can still survive. And you can then look through this window and see how structures form on the wing of the butterfly. And the structures that we are after are these scales that you see in the background of this image. The scales that carry color, that carry different functions for wedding, that also are involved in sort of manipulating flow fields across the wing to help with flying. And we want to see how these things form. Because if we see how these things form, we hopefully can also understand the biomechanical processes, mechanics here is important, that drive the formation, and maybe we can copy that or use that. So that work, that imaging, what happens there, and this image that you can see right now here, a cross-section through different scales done by an optical technique that's called reflection phase microscopy. We are seeing in the in vivo butterfly how these scales form. Here you see a sort of progression in the bottom left of the patterns, the scales as they form in the butterfly, and the structures that we're interested in, ridges, scales, and features. And Anthony is doing this imaging together with people from Peter So's group, Sang-Soo Kang and Sahid Yaqub, who help us really a lot on the optical side. Now, what we ultimately hope to be able to learn is how these structures that you can see in the slide, these incredible sort of sophisticated architectures form. The butterfly that we study is this Vanessa Cardi, the center stage one here. But there's a lot of other ones that change the morphology of these scales and the structure of it. And those morphological changes, I think, are all sort of part of a playbook, of a cookbook, if you want, in the natural system. And if you understand the most generic architecture of the Vanessa Cardi butterfly, maybe you have a handle on understanding these more colorful ones, the more sophisticated ones, the things that form fibers, or that sort of do things that are diffraction type architectures. So there's the hope. I hope I can convince a lot of people that this is worthwhile doing. We're starting modeling now. My student Amina Matt, together with Greg Carter, and also Jan Tots, a postdoc in my group, and in Jan Dunkel's group, they're studying the modeling aspect of this now. They're trying to get behind the mechanics and how we can actually model what happens in the formation of these sophisticated cuticle architectures. Now I have at least a small fan group for this, as you can see here. When the whole shutdown happened, I managed to convince Anthony to let me have a couple of his butterflies. And we have been hatching and raising them since at home. And here are two boys that sort of benefit most of that, including me, of course, as well. My two sons, Leo and Eric, that get quite a bit of kick out of this. And I get a bit of free time with homeschooling. All right. This brings me to the main course now. I want to speak more about sort of the bio-inspired materials that we make. I already mentioned the part on the butterfly-inspired light-emitting surfaces as a bit of a palette teaser. And I'm going to talk more about color-tunable photonic fibers now, and also time permitting about these color-changing droplets. Am I about the right speaking speed, or do I get carried away? Cool. Thank you, Teresa. All right. Good. Well, before we start on any of the particular projects, I want to sort of do a more generic little teaching session on what is the key component of light manipulation that we use. What we do is we split light, as you can see with this rather crude axe. We delay some of that light, and then we recombine it. There is a really beautiful device that can very elegantly show the concept behind this, and this is a MechE interferometer. It consists of two beam splitters and two mirrors. And when we send light, in this case, I used red color, so wavelengths about 700 nanometers, and obviously the length scale here is really off. When we send light with a certain amplitude, with a certain electromagnetic field, and you can see at the bottom here, the beam splitter, the first one, will split that into two different components. The first one propagates through and then gets sent by the mirror to the next beam splitter. And the other component propagates upwards in this sketch here, and then gets sent to the other beam splitter as well. We can tune the beam splitters, it's 50-50, so we can play on the amplitude to be even. And depending on how these two waves then come back together here, they go either out in full force, come on, here, and that means on the other side, on the other pass, I can't get much. So there's an either or thing here that I can play in some sense, but I can also play amplitudes in between. Now, what happens if I pull this back a little bit and we put another medium in one of the arms of the MechE, which is balanced interferometer? Then the first arm is not going to be affected, but the second arm, there we squeeze the wavelengths a little bit. We sort of just push those wavelengths together. We change the speed of light, and that means we delay the second wave with respect to the first one, and that can now lead to the fact that they interfere destructively on the one output, and that means I get the light on the other output. So in essence, I choose, by just choosing what material the light goes through, where it ultimately is up. I'm splitting it, and I define where it's recombining and where it's going. I can play the same trick with a different color, and here now I have that sort of mass of different material in the upper arm, and so the lower arm is not affected, but the upper arm is squeezed again. But for green now, the squeezing might be such that actually things come in constructively here and interfere on this output and not on this output. And so then, with this block of material in the upper arm, I will have chosen to have green go to the right and red go to the top. I have separated my colors. And that's the whole concept, playing with the material properties, delaying light, reflecting light of different interfaces, in this case, these two beam splitters, that we play over and over again in our research to sort of control how light is manipulated. All right, that's a preload. And I'm going to the first system where we play this trick. And these are the colorful fibers that Joseph is working on in my group, Joseph Sand. This is inspired by another group in Exeter University in England, Pete Vukusic and Alfred Ledgebridge, a long while ago now, 2013 or so, was where we wrote first about this work, from this blue fruit. This blue fruit has beautiful long cells on its surface, and these long cells are stacked in several patterns, but the intriguing aspect is that when you look into such a cell, it's full of stuff. It's a bit like a toilet paper roll, different layers stacked up on each other, and these layers are very regular. And here, in some sense, it's not quite a true picture, but in some sense, you can argue about these black and dark, dark and bright sort of contrasts are the different interfaces where I can split light, and the different layers where I can delay light in the analogy to my interferometer that I showed you before. So the simplest manifestation of that effect that creates the color blue in the fruit that we could think of was to create fibers where we roll multi-layers around the core. And if we use the right materials, these soft and fluid metal with which we like to cook, as I told you in the beginning, we can actually deform these materials, deform the optical interfaces and the spacing between them, and that allows us to change color. And that has been quite a lucrative sort of pastime for my group that has gotten some good interest in the last couple of years. The important part here to see is what changes color is that all these different layers, as I'm drawing them here, you can see how light comes in on the top, and there's 3 different beams that I'm sort of interfering, if you want. If I now change the spacing between these layers, this is the handle that we have, I can change the color that comes out of these fibers. That means squishing, pulling, deforming it makes the color of these fibers change, just because I change my interferometric setup, if you want. All right, you see a microscopic image of this on the left here. And on the right, you see a representation that we like to use quite often because it's intuitive. It's sort of the CAE color space, the space that represents how humans can see different colors, and the space that you can use to transfer a spectrum, intensity versus wavelengths representation of the light that you perceive in these color tones. This horseshoe type form puts blue and red and green on opposite corners. And depending on how far you are out on the edges, the more pure the color is. So the purest colors are on the outside edges. We can model the fiber behavior because you know this layered architecture's optics very well. And you can see the optical modeling data in the center, or in this part of the image that I'm showing from a pointer. And here you can see a fiber that Joseph has conceived after the model. And that matches very closely. So we can really control this and design these fibers according to the color trajectories that we want to have. In this case, it goes from green to red as you strain it. All right, and then what Joseph has done is he built a whole setup that you see in the top left corner, top right corner here, that can combine optical measurement with mechanical measurement. And the data set that you see here is quite full, but it's not too complicated to understand. What we do is we plot the strain that he controls at the top. We plot the stress that he measures with a force gauge on these fibers. So he just has something that measures the force at the end of the fiber. And here we plot the reflection peak intensity. That's a strong sort of spectral feature where the colors are sitting in this color map. So the red bar is the important part. I stretch out the first cycle just for you to see that it's quite linear. And then you see cycle 10, cycle 100, and cycle 1,000 and 10,000 of this performance. And that means you can actually play with these fibers quite a bit. They're pretty robust. And here is a bit of additional data that shows you the color plot again in the center, the CAE color space, and how this color trajectory stacks up for the fibers. But you also see the change in wavelengths, and that's very robust. We get sort of 2 to 4 nanometers variation over the whole 10,000 cycles in the peak position for any given strain. And here you see the reflectivity. And there's a little drop at cycle 10,000. So there is something of a phenomenon that we have to figure out exactly what happens there in terms of aging of these fibers. But when you look at the reflectivity, this is 0.7%. We're not showing this towards 0. We're showing this between 70% and 100% reflectivity. And that's bright. That's brighter than any of your pigments in your shirts or in your environment at home. All right. Now, these fibers have proven rather useful lately. One of the really cool things that came out of this was in collaboration with Vishal Patil and Jon Dunkel in the mass department. And that's all about tying knots. It's all about these very simple, if you want, pastimes that as a sailor or as a climber or as a surgeon, you would be very fond of, because these are sort of métiers and businesses where knots become quite useful, powerful, if you want. And you can see me tying a reef knot up there. And here is a fiber that Joseph tied a knot into, and he's pulling on that knot. He's sort of tightening down on it. And you will see that the colors change in this. Now this became really interesting to Jörn and Vishal, because they can model knot mechanics. And they came up with a very nice mechanical model first that they ran by simulations, but then they built a topological model on top of this. And here you can see 4 different knot types. The reef knot that I'm tying in the top left corner, that's the one you want to tie. That's the best knot of this family of 4. The granny knot, often a shoelace knot as well if you want, and then the thief and the grief knot. To some extent, the names of these knots are actually programmed. The grief knot is a really bad knot. That's not something you want to hang something off, especially yourself from, if you climb or something like this. The thief knot is actually a knot that sailors used to tie because it's very similar to a reef knot. And so they would know if somebody untied their bag, if at the end of the day when they came back, there was a reef knot instead of thief knot in there. The reef knot is the best and the sort of strongest knot in this context. And Jörn and Vishal have figured this out by just defining 3 different topological parameters. The number of crossings, it is n. Then the number, it is circulation. And the other one is sort of, this is this thing here. And then tau is the number of sort of plus minus crossings. Am I getting this right? I always mix two up. So let me actually make sure. This is the nicest thing about this whole online thing. I can go into my paper and look this up very quickly. Now I got it again. So we have n the number of crossing, tau being the circulation. You can still see my screen. Gamma being the circulation, and tau being the twist fluctuation. That meaning essentially whether the one crossing rotates the fiber in this direction or rotates it in the other direction. And these three topological parameters are sufficient to explain the relative strengths of these different knots. So it's a really beautiful, powerful, simple model that allows you now to distinguish between different classes of knots and different families of knots within the same class, if you want, and to figure out which one is actually stronger and a good one. So here are a couple of different knots that you can see tied by Joseph in the experiments. And this is a figure of 8 knot. This is a simple trefoil on the right. And then Joseph and Pichal together did a prison escape experiment where they tied these knots with relatively slippery loop and then put the weight on this and saw how fast these knots unraveled. And in parallel, Vishal would model these knots via his mechanical simulation, but also via the topological model. And you can see in all this data here, for all these different types of knots, everything falls nicely on top of each other. Data points are these small circuits, full circuits, and then different models are these different triangles and big circuits. So this topological model actually, in combination with showing the color that's related to strain and stress in these fibers, helped us to really understand knot behavior. And that could potentially be useful for sailing to get the right knots done, but also for surgery. We have a bit of interest now of people that look at the closure of wounds with sutures and what knots could do there in this context. Now, there's one other point about these fibers that I'm really excited about, and this is that you can also use them to do something useful in the medical context, not only the surgeon knots, but also for us, what became pretty evident after discussing with doctors at Brigham and Women's and in other contexts is that measuring pressure in many medical contexts is not that easy. One big one is the simple one of just applying a bandage. So if you apply a bandage on your arm or another person's arm or leg, you kind of guess the pressure that you really apply. And there are features in some bandages to help you with that guessing, but on the whole, it's still something that is very arbitrary to some extent. And then there's conditions like venous ulcers, so sores on the leg of more elderly patients, or sores on people's legs when they have to be in bed because they're paraplegic for different reasons, where the pressure has to be quite controlled in this range of 30 to 50 millimeter mercury. Now, we know that we can put these fibers into bandages by just sewing them in. This is easy. And we also know that the pressure of a bandage that's elastic depends on the strain that you apply. And the strain is the thing that ties fiber color to fiber strain. And that means we have a direct tie between color of the fiber and strain to strain of the bandage and pressure of the bandage that you exert on a person's leg or arm. And what you can see here is a bandage with fibers in it. In the top is one fiber that we zoom in on. The bottom graph shows you the peak position, the color as a function of pressure, and we tune that fiber to be yellow in the right pressure range. So this works. The pressure of the fiber can be encoded in the color of this, or the pressure of the bandage, I should say, can be encoded in the color of the fibers. And here you can see what Dan Joseph, together with Marie, who was a research scientist in my lab, have done to show that concept, at least on the lab scale, at least with us, people that don't know a lot about how to apply bandages. Since we don't have all that training in applying bandages, you can see that the control cases, these top two cases, the gray and light gray ones, are not quite on target. It's not that easy to get the pressure right that you want to have on ankle, mid-calf, or under the knee. But with a good color chart and a colorful bandage, you can actually get these pressures a bit more controlled. The distribution here is narrower, and we hit the target pressures pretty well in these initial sets. And now we're working with people at Brigham and Women's to show that this also works in the clinical context. There was always a caveat on this, and that was that we made these fibers with a technique that is lab-based and not necessarily scalable to the highest extent. We spent a long time figuring out how to scale this to get actually to that point here where we can have fiber in the whole bandage, right? Not just a few pieces of fibers distributed by the bandage, but having the fiber in the whole bandage, or even making a whole bandage out of these fibers if you wanted to. What made the switch here was that at some point, Ben Miller, a grad student in my group, made a really cool invention, if you want, or really cool discovery, rediscovery of an old technique, Lippmann photography, and use it for creating colorful surfaces that change their color when you deform them. So what you see here is when playing with a sheet, it's about the size of half a credit card if you want, and you can now make sheets of the size of tablets or business books, and he does it with an optical technique, just a bit of light exposure, and then it's done. So this is something scalable. And that means with this technique, we can make whole bandages out of this material, or shoes or textiles or whatever you can think of. So that sort of, I think, is breaking that scaling problem that we had for a while. This is the soft materials, the elastomeric one and the stretchable ones. And I want to push this a little bit further. I want to go full into the softest stuff that you can think of, fluids, things that deform very easily with very little force. And that's work that is led by Sarah Nagelberg in my group. And she does it together with a lot of different collaborators. In my group, Hannah Feldstein, Jan Tots, also Amina is contributing to some extent. And then with other groups, Amy Goodling in Penn State, together with Lawrence Sarsa, and also colleagues, faculty colleagues at MIT, like Tim and George, and you can see here. And this is still fully in the realm of colors. You can see how colorful these droplets are. And I want to show you an effect that you can actually try at home. This is a hot plate with a petri dish on it. There's just water in there. You heat that water up a little bit, and then you take another plastic surface, like a petri dish lid or a Tupperware box if you want to, and you can see how you create these colors. So it's a very simple thing, and I think many people have seen this before. We were lucky that nobody had really explained that, and by the means of these droplets that Lauren and Amy can make so wonderfully and that we work with, we figured out how this color gets generated. And here you see the early data. You see a side view of a droplet, and you can see the first pictures that Amy and Lauren sent to Sarah and me to show that these sort of droplets that are made from two different oils, slightly different refractive index, and this really nice internal interface. The size of these droplets are about 100 micrometer. These droplets can generate colors under the right conditions. These were the first pictures, and then they showed us these images a while later. This is a video that should hopefully play. What you see is a petri dish of these droplets. As I said, they're oil emulsions in water. The size of the droplets is pretty monosparse, so they're all about the same size. And the light incidence angle and the orientation of the sample doesn't change. What we change is the observation direction. So you move the camera around or your head by that matter. And you can see all the different colors that you get over there. That, by the way, was what I was brewing in this kettle. Now, the question to ask is, why do we see these colors? And I believe I have about 5 minutes left, right? Perfect. Okay, cool. Why do we see these colors? This was sort of a simple question that could come out of this. And I had a simple and wrong answer in the beginning. We thought that was the rainbow. I might have been the one who saw it very strongly. I think Sarah was suspicious of this. She was smaller than I was. And the rainbow was completely wrong. When you model the rainbow, you don't get to these colors as hard as we tried this. So then we had to start from scratch, essentially. This problem became a lot more interesting. Also took us a while to figure this out. But we sort of started detective work, and we started to ask, what on earth is that? How do we characterize this? And we went through any possible optical phenomena that we could figure out on interfaces. Turned out to be rather simple at the end. But if I would give you the answer right now, that wouldn't be too much fun. So I'll leave you a little bit alone. I'll tell you how we found that. And a mix of historical and pedagogical, if you want. So first thing that we saw is that when you look at these microscope images that you see here, still the same droplets, two different phases, high index phase at the top, low index phase at the bottom, light comes from the top in the microscope. You see color only from the rings, only from the edges forming these rings. When the droplets are the same size, you get the same color, yellow on the left here. If they're different size, you get different colors. So it's a size dependent effect. And the edge is quite important. The edge actually tells us something about how light has to propagate through there. You don't get color from anywhere else from the droplet. The way the light has to propagate through there to create this edge effect is by total internal reflection. And so what I'm showing here in brown is this internal interface of the droplet, and a light ray coming in from the left and bouncing around this interface until it comes back out on the right, and then you can see it. So that was something we figured out pretty quickly. But total internal reflection doesn't tell you anything about how color could be changed, right? We're just redirecting light. We're not splitting it up, and we're not sort of recombining it. And that's sort of the key that I want to point out here. What's splitting, what's recombining here. And that took us a while to figure this out. One thing that worked for us was this sort of ping pong ball imaging. So what you see here is what we can call scatteroscope or ping pong ball microscope. And what we do is we have a small half sphere, semi-translucent. The ping pong ball is really good for that. Send light through a hole into it, and then we capture the reflected light on the whole surface of that semi-sphere. So you can see this here in real. On the left, we have an image with the droplet carpet in the petri dish, the ping pong ball in the middle, and the lights in the room are still on. And when we switch off the lights in the room, you can see all the colors that these droplets send into different directions. And here's a couple of pictures that come from these ping pong ball scatterograms, if you want, for different increasing droplet sizes, but also for changing curvature. So we really sort of decide where light goes for a given color into sort of which angular direction in space. Doesn't really explain yet what happens there. For that, we really have to look very carefully at what happens in the droplet again. And these sort of data sets that I show you very big here helped us understand this. We could ask about why are there dark spots? Why are there spots where you can see rainbows? And why are there sort of bands that interpenetrate each other? The dark spots are starting to become really easy when you think a bit about it. One is you need total reflection. So if your light beam that's shown on the left comes in at a too low angle of incidence, it will just transmit through the interface. And you can also not do the things that I show in the center and on the right. You can't, for a given angle of incidence and a given angle of observation, get light into certain directions. So that explains all these dark spots that you see in this part here. There's only sort of very specific directions in which you can send light. That still doesn't explain why we see colors. We still haven't sort of recombined something and shifted something, delayed something. And for that, it was very important to figure out that actually there is a quantity for each optical, for this optical path, that's the path length. So we can delay the phase of the wave by changing that path length. And then the other important part was to actually figure out that there's not only one path that light can take through this droplet, but there's many different reflection paths that you can take to bounce from one incidence direction to the other observation direction that you can see there. And this is where the splitting comes in. We split the light into these different paths, and each path gets a little bit of a different shift. And then when you combine them on the outgoing side again, you can write a simple formula down that accounts for the phase change up on reflection, this thing that I put my pointer around here. It accounts for the phase shift, this is this complex exponential, for the whole path length of each path. And then we just sum over these paths. And since we measure intensity, not amplitude, we just square this up and get our reflectivity. And when you model it this way, what you get is a model color distribution that you see on the right. And this is all work that Sarah has done. And you can compare this to the measured color distribution, these ping pong ball experiments on the left. And it's really nicely, strikingly similar. And the sort of the different paths you can also, we could have seen this in the beginning, you could have maybe thought about these rings as sort of the indication of slightly different bounce paths, but we didn't. So we took a longer time to figure this out. All right. This works very simply in this sort of experiment that I showed you with the condensation of droplets on the surface. You can see water droplets condensed on a PDMS surface. And you can also change whether you see colors or not just by changing the hydrophobicity of the surface. That changes the curvature of this droplet interface. And it either allows light to be in this sort of total internal reflection interference mode, or it doesn't allow it. So you can control this. And there's a whole lot of cool things that you can do with these droplets. You can use them with lenses. You can use them to generate color where there is intrinsically transparent materials. And you can also use them to sense pathogens. And I haven't mentioned any of that, but that's work with Tim. That's really exciting because there these small droplets act on small scale chemical stuff that we can read out macro scale. All right, I'm wrapping up here. The key things that I wanted to bring across is that there is a whole lot of things that you can learn by looking at nature and seeing how nature forms optical materials. Soft and fluid matter are really powerful. It's underutilized in optics, but if you use them right and we control the structure, then we can do a whole lot of really interesting wave optics to generate color, to generate interesting optical effects, and to use it for applications to some extent. All right, final slide. All the people that have worked on this and more, most people I have mentioned already, but I think I want to point out Joseph and Sarah and Ben and Anthony in particular because I showed all their work. Cecile's work I haven't really discussed, so you can ask questions about this if you want to. And I've been very, very much inspired by a lot of really bright faculty that you can see in the second row here. And I want to thank you for all your attention, and I hope I'm fairly well within time.
Mathias, thank you very much. That was a very colorful talk. Excuse the dad joke.
Thank you.
So I'd like to turn it over to the participants now. Please use the Q&A feature and feel free to ask some questions, and we'll try to take them in the order in which they come. I will kick it off with one question that I had in terms of as you look at some of the other applications for looking at thin films and how color is produced beyond medical. I mean, how are you working with sort of people outside of MIT and industry to identify applications that can be interesting?
This is a really good question. And the answer is we're really still working this out, even after the time that I'm at MIT for a while now. I'm working largely based on the fact that we come up with cool ideas, and then we get those ideas tested or rejected by people that actually work in this space. That's one way of doing this. And by that nature, I find more people that actually can use this. So for example, we have something that's starting to be more and more firming up with New Balance, where they think that our materials can be useful for their textiles. Whether this is in shoes or clothing is sort of up for debate, but these are the things. The discussions with the doctors, Mathias Kolle at Brigham and Women's, for example, that sort of honed us in on what are the features that we need in these fibers. The color change is nice, and you can have a color pattern or a color chart if you want along with this, but more binary features, if you want, could be very useful for them. It's easier to see and quicker to find. So they guide our designs, right? And then writing grants, surprisingly, has honed me on really interesting things, because you see an opportunity coming along, and together with my students, we ideate on what is possible, and then we sometimes sort of put together grants that actually work, and sometimes they don't work, but we're still excited about the idea. Right? You touch on a thing that I could need help with actually, is sort of figuring out how companies do things. A good company that I think would be very beneficial to work with is 3M, but they're also very difficult to read in terms of when they're ready to go with something, right? So learning the metrics, learning this sort of interaction with company professionals and getting what they need to them is an interesting challenge. And I'm playing a lot on guessing there as well.
But it's always it's conversations and talking a lot to figure those things out. So we have some questions from the audience. So the first one coming in was, were the droplet principles the inspiration for your recently announced dark field microscope adapter?
Well, the droplet lenses, good. The droplet lenses for this microscope, for the SLAT imaging, I think this is the question that goes to here. So let me share the screens again, and then I'll ask if I'm correct with this. I don't want to have the wrong speculation. But the question was if the droplet lenses are the inspiration for these sort of imaging techniques. Is that correct?
Yes, I think were the droplet principles the inspiration for your published dark field microscope adapter?
Yeah. Yes and no. The initial inspiration was that we found these really cool butterfly structures a while ago on a wing of butterfly that create these very interesting color mixing effects. And initially, naively and not well informed, I thought we could build really cool lasers out of this. So you have all these small little cavities that you see in the center picture. And if you get them to emit light in a certain wavelength, and you have the right sources there, then you can get coherent emission between these different cavities that you see also in the microscope picture on the right. And by switching them on individually, you might be able to steer where the light goes and what sort of spectral nature it has to some extent. With quantum dots, that doesn't work because coherent sort of emission by quantum dots is not that easy, and I don't have the tools in my lab for this. The lenses and all this sort of thinking about the frequency space, part of the things that I learned from George actually a lot in this course and in the actions, George Papastatis, that helped me with the optics, with the understanding of thinking about, hey, what is the imaging modality here? What is the usefulness, right? And so when we blatantly failed on the laser stuff, what was really exciting to come out of this was this hollow cone of light emission from these surfaces. With this design of a concave reflector, an emitting layer that creates the light, and the Bragg mirror that is a gatekeeper that allows photons or light only coming out at certain angles, we can tailor the emission angle, and that is really useful in the dark field microscopy field area, where you want to tailor these emission angles. You can see very transparent materials very easily with that if they scatter just a little bit, and as you can see at the bottom here. The lenses, and this whole going forth and back with Sarah and a lot of other colleagues on the optics, I think that got me ready to actually think about this partial current imaging mode that we see there. So it's been an evolution, seeing this stuff because I had this background of the lenses.
Very good, thank you. I think we have time for one more question. What time scales do the color change responses happen with your materials, and what kinds of dynamic color changing systems can they be used for?
Great question. It's all mechanical. Think of this as a spring. It's almost like the deformation in the fibers. It's almost like you can write down stress is equal to an elastic modulus times the deformation, right? So there's no phase lag in that, unless you go to really high frequencies. So when you push on this material, you get the color right away for the deformation that you create on that time scale. So I think we haven't pushed any limit in terms of finding out where at which point we create sort of a lag in response. For the droplets, that depends on how quickly you can switch dose. And that's an interesting question because now you work on fluids. And I haven't said very little about how you can change the conformation of these droplets, but you can, for example, change the surfactants. I have not brought the slide with me, unfortunately, but I can hand wave it, I guess, on a picture of a droplet. So you see in this top corner here, you see that droplet interface. And we adjust that in that position or in any other position that we want by changing the surfactants that we have in the surrounding. There's one phase, this is often a heptane or a hexane, and this could be fluorohexane. There's two different types of surfactants, and by changing the ratio, we can change these conformations. But you have to get the surfactant there. So that would define the speed of switching. What you also can do is you can put a surfactant in there that is light responsive. And so when that switches its conformation and changes the surfactant strengths, then it's the switching of that surfactant that defines the speed. And that can be in a couple of microseconds. And the other thing that you could do is, and we're playing with this, Sarah Nagelberg has done the experiments, Jan has done the modeling on this, Jan Tots. We are driving these thermally. You can move these droplets around by thermal gradients. They're very responsive. And I think that's a millisecond time scale behavior. So there's a huge range of time scales depending on what stimulus you use.
Very good. Thank you very much. So again, Mathias, thank you very much. A wonderful talk. I know there's a virtual clapping, but I'm sort of quietly clapping here. And let me just share my screen very briefly here to announce. So next week on Thursday instead of Wednesday, we'll have Linan Zhang, who is a doctoral candidate in mechanical engineering, working in the group of Professor Evelyn Wang. Sharing his work on ultra-high efficiency desalination via a thermally localized multi-stage solar still. So I do encourage you to spread the word to your colleagues and please join us for that next talk. And I guess as my screen goes there. So we're going to try to cycle through faculty talks, student and postdoc talks, and external to MIT talks, so including national labs, our industry partners. So please, if you are interested in sharing your expertise, your work, the projects that you're working on, either for research or product development out in industry or in national labs, do please let us know. Or if you're at MIT, do let us know as well. And we look forward to inviting you back. And please stay safe, stay well. And again, Mathias, thank you to you and to your team and everybody else. Have a good day. Thank you.
Thank you, Brian.
Thank you. Nice to see everyone.
Thank you. Have a good day, everyone.